Welcome to Easy Microbiology.
Good morning, everyone. Today, we will be discussing Campylobacter coli, a gram-negative bacteria. We will be exploring its habitat, morphological characteristics, cultural and biochemical characteristics, host range, virulence factors, mechanisms of antibiotic resistance, and epidemiology. Please stay with us and follow the presentation. Let's get started. Campylobacter coli. It is important to note that this bacterium can cause gastroenteritis in humans and shares similarities with C. jejuni in terms of its transmission, symptoms, and clinical presentation. It is important to take necessary precautions to prevent its transmission and seek medical attention if symptoms arise. C. coli. This bacterium is commonly found in the intestines of animals, especially pigs, cattle, and other livestock. It can also be present in the environment, such as in water sources or on contaminated surfaces. Therefore, it is important to practice good hygiene and food safety measures to prevent its transmission. C. coli is a bacterium that has a curved or spiral shape, similar to C. jejuni. It has flagella, which are long, whip-like appendages that allow for motility or movement. These flagella help C. coli to colonize the intestinal lining, which can lead to infection and illness in humans. To culture C. coli, selective media such as Campylobacter blood-free selective agar or charcoal blood agar are used. These media contain specific nutrients that allow for the growth and survival of C. coli and inhibit the growth of other bacteria. Additionally, C. coli requires specific growth conditions, such as reduced oxygen and increased carbon dioxide levels, for optimal growth. This means that the culture plates must be incubated under microaerobic conditions, which provide the necessary environment for C. coli to grow. These conditions are important for the accurate identification and diagnosis of C. coli infections in humans. C. coli is a bacterium that is catalase positive and oxidase positive, meaning it produces the respective enzymes. It is also microaerophilic, which means it prefers low oxygen concentrations for growth. Similar to C. jejuni, it is non-fermentative and does not utilize carbohydrates for energy production. Instead, it uses other metabolic pathways to obtain energy. These characteristics are important for the accurate identification and diagnosis of C. coli infections in humans. C. coli is a bacterium that primarily infects animals, especially pigs and other livestock. However, it can also infect humans, leading to gastroenteritis. This can occur through the consumption of contaminated food or water, or through contact with contaminated surfaces. In humans, C. coli can cause symptoms such as diarrhea, abdominal pain, fever, and vomiting. It is important to practice good hygiene and food safety measures to prevent its transmission and seek medical attention if symptoms arise. C. coli is a bacterium that produces various virulence factors, which are molecules that contribute to its ability to cause disease. These virulence factors include adhesins, invasion proteins, toxins, such as cytolethal distending toxin and flagella. Adhesins and invasion proteins help C. coli to attach to and invade the intestinal lining, while toxins and flagella contribute to its ability to cause inflammation and induce cellular damage. These factors work together to enable C. coli to colonize the intestinal lining and cause gastroenteritis in humans. C. coli, similar to C. jejuni, can develop antibiotic resistance through acquired resistance genes or mutations in target sites. Acquired resistance genes can be transferred between bacteria, allowing for the spread of resistance. Mutations in target sites can also lead to resistance by altering the structure or function of the target site, making it less susceptible to antibiotics. This resistance can lead to reduced susceptibility or complete resistance to certain antibiotics, impacting treatment options. It is important to use antibiotics judiciously and to follow appropriate antibiotic stewardship practices to minimize the development and spread of antibiotic resistance. C. coli is an important cause of gastroenteritis in humans and is often associated with the consumption of contaminated food, such as undercooked meat or unpasteurized dairy products. This can occur when the food is contaminated with C. coli from animal sources, such as pigs or cattle. In addition, infection can also occur through contact with animals or contaminated environmental sources, such as water or soil. Symptoms of C. coli infection in humans can include diarrhea, abdominal pain, fever, and vomiting. It is important to practice good hygiene and food safety measures to prevent its transmission and seek medical attention if symptoms arise. Upon ingestion, C. coli colonizes the intestinal lining, which can lead to inflammation and damage to the epithelial cells. This can result in symptoms such as diarrhea, abdominal pain, and fever, which are similar to other Campylobacter infections. The severity of symptoms can vary, ranging from mild to severe, and can last for several days to a week. In some cases, C. coli infection can lead to more serious complications, such as Guillain-Barré syndrome, which is a rare but serious neurological disorder. It is important to seek medical attention if symptoms arise, especially if they are severe or persistent. C. coli infection elicits both innate and adaptive immune responses in the host. The innate immune response is the first line of defense against infection and involves the activation of immune cells such as neutrophils and macrophages. This response is followed by the adaptive immune response, which involves the activation of B cells and T cells to produce antibodies and eliminate the pathogen. Antibodies, particularly IgA, play a role in limiting bacterial colonization and neutralizing toxins. IgA antibodies are produced in response to C. coli infection and are found in the intestinal lining, where they can bind to and neutralize the bacteria and its toxins. This helps to limit the spread of infection and prevent further damage to the intestinal lining. The immune response to C. coli infection is important for the clearance of the bacteria and the resolution of symptoms. Laboratory diagnosis of C. coli infection involves collecting stool samples from the patient and testing them using culture methods, such as selective agar plates. These culture methods involve growing the bacteria in a selective media, such as Campylobacter blood-free selective agar or charcoal blood agar, under microaerobic conditions. This allows for the accurate identification of C. coli and other Campylobacter species. In addition to culture methods, molecular techniques such as PCR can also be used for detection. PCR can detect the DNA of C. coli in stool samples, allowing for a rapid and sensitive diagnosis. These molecular techniques are particularly useful in cases where the bacteria may be difficult to culture, such as in patients who have recently received antibiotics. The laboratory diagnosis of C. coli infection is important for the accurate identification and treatment of the infection. Prevention of C. coli infection involves practicing good food hygiene, such as thorough cooking of meat and proper handling of raw food. This includes cooking meat to a safe temperature, washing hands and surfaces that come into contact with raw meat, and avoiding cross-contamination between raw and cooked foods. In addition to good food hygiene practices, general hygiene practices, such as hand washing, are crucial in reducing the risk of transmission. This includes washing hands with soap and water before handling food, after using the toilet, and after contact with animals or their environment. It is also important to avoid consuming unpasteurized dairy products and to drink clean and safe water. By practicing good hygiene and food safety measures, the risk of C. coli infection can be greatly reduced. In most cases, C. coli infections are self-limiting and do not require specific treatment. However, severe cases or those in high-risk individuals may benefit from antibiotic therapy. Macrolides, such as erythromycin, and fluoroquinolones, such as ciprofloxacin, are the drugs of choice for the treatment of C. coli infections. The decision to use antibiotics should be based on the severity of symptoms, the risk of complications, and the susceptibility of the bacteria to the antibiotic. It is important to use antibiotics judiciously and to follow appropriate antibiotic stewardship practices to minimize the development and spread of antibiotic resistance. In addition to antibiotic therapy, supportive care, such as rehydration, may be necessary to manage symptoms. Campylobacter coli is a gram-negative bacterium that causes gastroenteritis in humans, primarily associated with the consumption of contaminated food. Understanding its characteristics, epidemiology, pathogenesis, diagnosis, prevention, and treatment is essential for effective management and control of C. coli infections. C. coli shares many similarities with C. jejuni in terms of its transmission, symptoms, and clinical presentation. It produces various virulence factors, including adhesins, invasion proteins, toxins, and flagella, which contribute to its ability to colonize the intestinal lining, cause inflammation, and induce cellular damage. Laboratory diagnosis of C. coli infection involves collecting stool samples and testing them using culture methods, such as selective agar plates, or molecular techniques, such as PCR. Prevention of C. coli infection involves practicing good food hygiene, such as thorough cooking of meat and proper handling of raw food, and general hygiene practices such as hand washing. In most cases, C. coli infections are self-limiting and do not require specific treatment. However, severe cases or those in high-risk individuals may benefit from antibiotic therapy, with macrolides or fluoroquinolones being the drugs of choice. It is important to use antibiotics judiciously and to follow appropriate antibiotic stewardship practices to minimize the development and spread of antibiotic resistance. Thank you so much for tuning in today. I hope you found video insightful and enjoyable. If you did, don't forget to give it a thumbs up. It really helps support the channel.